I'm super excited to share one of our latest work on nucleic acid therapeutics. Our group work with short interfering RNA. These are precision medicine drugs that can specifically downregulate expression of proteins of interest. So what we actually were trying to do was to figure out a new method to make these drug molecules more effective. So the way we do this is by modifying these sequences with specific chemical modifications. Specifically, we developed 2' hydroxy modifications, diol modifications that we incorporate into the sequence. So we do chemical modification as well as structural modifications. So one of the main advantages of this design is that you can control the activity of the sequence and can take away the side effects of the sequence. So usually these drugs are very specific, but sometimes you get non-specific interactions, non-specific downregulation of undesired proteins. So to make it specific with these modifications, you can inhibit the two major side effects that comes from microRNA-like effects and recruitment of sense strand, the non-functional strand, into the active risk complex that is the main factory where these gene silencing happen. So with our design, we can tune these properties and make these drugs way more specific than what it is known today.
Hi, in this paper, we have been working on chemical and structural modifications on siRNA to improve its activity. Traditionally, siRNA consists of two strands, sense strand and antisense strand, where antisense strand is completely complementary to the target messenger RNA, which is responsible for protein knockdown through RNA interference. Specifically, there is a region in antisense strand called seed region from 2 to 8 nucleotides from 5' end, which is responsible for mRNA recognition and RNAi activity. In our work, we have chemically modified the seed region with a 2' diol chemical modification through a phosphoramidite chemistry. Additionally, together with chemical modifications, we have also structurally modified the sense strand with 3 extra deoxythymidine nucleotides on the 3' overhang. When we introduced these chemical and structural modification into the cells, what we observed was the structural modifications on the sense strand improved the antisense loading into the RISC complex, where antisense strand is main active component of the cell in RNAi therapy. And when we have our chemical modification in the seed region, which could decrease the microRNA-like off-target effects, and when we combined these structural and chemical modifications, we could see a potential decrease in sense strand mediated off-target effects and microRNA-like off-target effects. If you want to know more about our work or want to collaborate with us, don't hesitate to contact us, and the link for the paper is given below.